Assalamu alaikum. Hello, my dear students. How are you? I hope you are well. Everybody becomes delighted when we think of our childhood. From the little one, slowly you have grown. This has been possible by mitosis cell division. Before this division, a cell take up preparation for the division. In this preparation stage, chromosomes of a cell become double after DNA replication. My dear students, our today's topic is DNA replication. Today we will learn types of replication and description of semi-conservative replication. My dear students, the process by which a new DNA is formed from old DNA is known as replication. This process takes place at the interphase stage or preparation stage of cell division. My dear students, now types of replication. Three models of DNA replication has been suggested by the scientists Levianthal and Crane. In 1956, these are conservative replication, semi-conservative replication, and dispersive replication. My dear students, first one, conservative replication. Conservative replication produces one helix made entirely old and one helix made entirely of new DNA. According to this model, the entire original DNA double helix acts as a template for a new DNA double helix, such that each DNA produces a completely new DNA double helix and another with a completely intact old DNA. Then dispersive replication, the replication by which the original DNA chain breaks and recombines in a random way and produces two copies of DNA composed of both original strand and new strand. Then semi-conservative replication. Semi-conservative replication produces two copies of DNA contain one old and one new DNA in each copies. So, the process by which two copies of DNA molecule are produced where each copy contains one original strand and one newly synthesized strand called semi-conservative replication. James D. Watson and Francis Crick in 1953 proposed the semi-conservative model of replication. Messelson Stahl in 1958 have proved semi-conservative replication of prokaryotic cells, and Howard Taylor in 1960 have proved semi-conservative replication of eukaryotic cell. Steps of semi-conservative replication. This process is completed by 4 consecutive stages. The stages are de-spiralization and breakage of hydrogen bonds, formation of replication fork, primer binding, elongation, and termination. At first, topoisomerase enzyme relaxes the DNA from its supercoiled nature. The enzyme helicase breaks the hydrogen bond between the bases of nucleotides of DNA and the double helix unwound to form single strand. The separation happens in the place where adenine and thymine bond present. Because there are only 2 bonds, that's easy to break. Single-stranded binding proteins bind to each DNA strand that prevent the double helix from re-annealing. SSBP stabilize single strand. The opening in the DNA are called replication bubbles or replication eyes. At either end of these bubbles is a replication fork, a Y-shaped region of the bubble. Replication begins at the site called origin of replication or ORI, where the DNA is unzipped when paired nitrogenous bases are separated. Replication occurs on multiple origins of DNA molecule. So, the separation of the two single strands of DNA creates a Y-shaped structure called replication fork. The two separated strands will act as a template for making the new DNA strand. Following this fork, RNA primase and then DNA polymerase begin to act in order to create a new complementary strand. Because these enzymes can only work in the 5' to 3' direction, the two unwound templates strand are replicated in different ways. Replication occurs separately on each template strand in antiparallel directions. One of the strands is oriented in the 3' to 5' direction, and RNA primer binds at the end of this strand and acts as the starting point of DNA synthesis. Then DNA polymerase added new complementary nucleotide bases continuously to this strand in the 5' to 3' direction. DNA polymerase can only add nucleotides continuously at the free 3' end and always form a new DNA strand in the 5' to 3' direction only. This continuously replicated new strand with 5' to 3' direction is known as leading strand. The other template strand of DNA is oriented in the 5' to 3' direction. Numerous RNA primer binds at various points of strand of this strand's 3' end. Primer acts as the starting point of DNA synthesis. Then DNA polymerase enzyme make DNA strand. This DNA strand is known as lagging strand. As the DNA molecule is very large, replication starts at different points at a time. Beside that, replication in lagging strands take place in small segments. After completion of a segment, the polymer enzyme is released. The different segments are joined by the enzyme ligase to form the complete strand. Thus, two new double-stranded DNA molecules are formed. At this time, RNA primer is released by the enzyme gyrase and the molecule re-spiralized. These two daughter DNA molecules contain one old and a new complementary strand. Thus the replication is called semi-conservative model. Importance of DNA replication. DNA replication needs to occur so that during cell division, new cells will also have a copy of organism's DNA. DNA is necessary to make all the RNA and proteins needed for cells carry out necessary reactions and cellular processes in order to survive. Semi-conservative nature of DNA replication ensures the specific traits that pass through generation to generation unchanged. Some enzymes needed in this process. Number 1, DNA helicase. This DNA helicase unwinds the DNA double helix at the replication fork. Then DNA polymerase. Polymerase builds a new helix DNA chain, DNA strand by adding nucleotides in the 5' to 3' direction. It also performs proofreading. Then single-stranded binding protein. This protein prevents the DNA double helix from re-annealing after DNA helices unwinds it. Topoisomerase. Topoisomerase enzyme relaxes the DNA from its supercoiled nature. DNA gyres anneal the semi-conservative strand and joins Okazaki fragments of the lagging strand. And primase provides a starting point.
Your homework is description of semi-conservative replication. Thank you all to stay with us.